welcome back. So we're going to restart, and we are having Devako with us. Dr. Devako, he will be starting with his session, and as you know, that both of these sessions will be appended with the hands-on session later on in the afternoon. So be prepared for that, ask questions, and you might have a very short session after this talk where some of us will be telling us about the opportunities that are available for PhD positions in Center for Computational Biology. Right? So let's get started.
Thank you, Ganesh. Good morning, everybody. My name is Debarka Sengupta, and I'm a computer scientist. I had nothing to do with biology, and I always thought when I was a child that biology is not really interesting. And that's the past now. Okay, I'm so much interested in biology, and I truly believe that there is nothing else which is more interesting than biology. It's not possible. Because there is a theoretical bound. You cannot be better than God, right? So you're studying what God has created. And some other guys are studying what man has created. Right? That's the difference. Alright, with that short introduction and to my subject, now I'll try to take you through a journey of the single cells, which is pretty new, and in India, it has not yet started. So we are trying to start those initiatives back in India as well. So to introduce myself, as I said, I'm a computer scientist. My PhD is in computer science, but accidentally I got to work in projects which were related to biology because DST was funding that project and it was my bread and butter. But later, so I initially worked on microRNAs, then I shifted to genomics. I was more working on the network aspects of microRNA and how microRNAs interact with transcription factors. And then onwards, after my PhD was over, I started working on in a lab of Genome Institute of Singapore. I started working on single-cell genomics, and that's when I got introduced to the field of genomics. Now, every field evolves, and so does the field of genomics. And possibly right now, I would rather say that it has undergone a tremendous revolution very silently with the advent of single cells. Because just think for a minute that whatever we have learned so far through this great tool that is genomics, we have always learned the properties of the features of millions of cells, isn't it? We have always learned about millions of cells because our assays are such that we cannot have the precision that we require to understand a single cell, right? So that's now possible with the single cell technologies because you can right now amplify the whole transcriptome and genome of a very tiny cell as an individual. So you can learn about its characteristics. And that's going to give you much more resolution into the biology that you're in, the field of the very aspect of biology you're interested in. So this is a fact, right? So conventional genomic assays basically teach us about the features of usually several millions of cells, whereas single cells really give you the window to see what is going on inside a single cell and what is the diversity, what is the heterogeneity. So big field, and it's pouring Nature and Science papers every day, okay, every week at least. So microarray, bulk RNA-seq, and DNA-seq are the kind of assays which we are more familiar with and which are being now challenged by single-cell assays. So you have, this is a bit costlier than all those bulk assays, but you are getting a lot of cells. The sample size is very huge, so it is very interesting from statistical perspective as well. So the more samples you have, it gives you more statistical power, right? So what you learn is actually very precise. And that's the beauty of having more samples, which is true for single cells. Any single cell assay will give you a lot of samples, at least hundreds of samples of the same. You can find many clusters and so on. So it gives you a lot of statistical power to analyze your data. So this is a very iconic image, okay? So why it is very risky to believe in a crowd? If you look at this figure without this circle, then you will all see that these all guys here are actually saluting Hitler. But this one guy here, whose name is August Landmacher, he is not. And that's exactly the problem of bulk assays, right? You always believe what you see on an average. And that's what has shaped our understanding of biology, right? But look at this guy. Now what happens if these are cells, right? And this guy is a cell, which is, let's say these are all cancer cells, okay? And these cancer cells can be cured through drugs, okay? Whereas this cell is a very stubborn kind of cell, which has certain properties, which if you don't know, you're not able to deal with. That's a common problem you can have in cancer biology. Can somebody shut this door? I'm getting distracted. So that's exactly the problem we're trying to solve with the single cell assays, and let's see what kind of applications we can have. But before that, let's understand what is the technology. And here's another example of heterogeneity. I was talking about cancer. And in cancer, whenever you do this kind of staining, very nice images that you get show you, it's not nice for the person, but as scientists, we always love this kind of pictures, right? Because you've seen the tremendous amount of heterogeneity. These are all breast cancer samples taken from 4 different individuals, and they look spectacularly different from each other. Now, just think about doing some kind of bulk assay, let's say bulk RNA sequencing, on this guy or this guy. You don't know what you learn. You learn something very average, so you will miss the diversity, right? So that is again a very compelling reason for looking into precision, looking at biology with very high precision. That's a need, right? We never realized that. We were so much occupied with the quick discoveries and the cost reduction of sequencing, we never, looked back, introspected, and thought about the fact that, hey, what I'm learning is actually an average property of millions of cells, right? So now, since we have, so this is a silent revolution, right? Now, some scientists actually realized that, and they kept on working on it, and they came with this breakthrough. And this is very recent. I'm talking about technologies which are just 3-4 years old. Okay? So when you say single cell sequencing, now for obvious reasons, you cover both DNA sequencing and RNA sequencing. Now DNA sequencing methods are a lot more costlier than single cell RNA sequencing. And that is one of the reasons for which, there are less DNA sequencing papers than RNA sequencing papers. It's a little challenging. The protocols are not that straightforward. And, so basically what you do is very simple, pictorially at least. So you have this tissue here, you are pricking cells, you're taking a lot of cells here as I have been saying so far. So you have loads of DNA, right? You don't have any lack of DNA because you can take as many number of cells as you want. And then you have loads of DNA which you can directly sequence, right? Whereas here, you are actually pricking into the tissue, you are isolating one single cell, and you are doing the amplification. So amplification plays a major role here. Now, there is a problem with the amplification. The moment you amplify, the amplification process is not 100% accurate, right? And it introduces huge amount of noise. As you amplify, the small amount of material, which is I think 1 to 10 picogram or something, which is very small amount, really small amount, and you want to amplify all the molecules that you have, so you're basically making so there's a mix of participants here. So some of you might be interested in the biology aspect, some of you might be interested in the computation aspect. So I'm trying to maintain a balance, but So let's see how it goes. So here you are doing the amplification. But the moment you do amplification, there are certain kinds of noisy signals that gets introduced to your data. And that's where we need the help of a statistician and the machine learning guy. Alright, so, and this, signal processing guy as well. Yes, so these are, topics which are very close to each other, but they're not exactly the same.
So yes. What kind of noise is present here which is absent in the earlier or the upper part?
So see, many kinds of noise. First of all, you are starting with a very small amount of material, right? So your dropouts will be more. Let's say one particular transcript fails to, you have many genes, right? Some of the genes are highly expressed, some of the genes are lowly expressed. So the lowly expressed genes are very small in quantity. So you may not end up amplifying them, right? So you may not get a copy of that at all. So what happens is that if you see the matrix, so finally what we start from is a matrix that we'll see in the hands-on session. So in the matrix, you'll see most of the cells in the matrix are actually zeros. That means you have not been able to detect that. That also, and in all the sort of noise sources that were already present with the bulk acids are still there. But you have some added noise because you're starting with a very small amount of material. So it's very hard to ensure that you are actually amplifying all the transcripts equally well. So that is the major source of noise. So you have a lot of zeros.
What is noise and what is information?
So the information is, alright, so is that, okay, so you have something expressed. I am fat, okay, somebody is thin, right? That doesn't mean he is not there. He still exists, right? So that's the noise in single cell. So I am fat. Now I am being detected as a very, moderately fat guy in single cell. Whereas the guy who is very thin, for example, my friend is a little, leaner than me, so he will not be detected. Isn't it a noise? And that, if you consider as the biology, then that will be a foolishness. That will make fool out of ourselves, right? So we are talking about, so I'm talking in general about, so I was actually, thank you for correcting me. I think I was talking more about RNA, right? So in DNA, I'm not really very aware of what goes in, I'm not very aware of the technologies related to DNA, but at least I can tell you for RNA, this is the problem. For DNA, I guess there will be similar kind of problems, but in DNA you at least have the single molecule, right? And you'll have to amplify it over and again. So many steps of amplification, PCR cycles you'll have to do, and those PCR cycles are not 100% accurate all the time. Right. So PCR, the noise that you get from PCR cycles is one, and then at the time of sequencing, since you have a lot of cells, you cannot do very deep sequencing, right? There's a problem with the single cells, right? You can invest a lot of money and you can do deep sequencing, and then you will get spectacular precision. But that's not always the case. We land, finally, when you do the sequencing, you get just a couple of millions of reads out of a single cell, because that's what we can afford in reality. So maybe it's different in DNA. It is surely different in DNA than RNA. But I was more talking about the RNA thing, which is the next. And
may I ask one more question?
Yes, please.
So how is it to identify a single cell which you want to isolate DNA or RNA?
How easy? No, it is not easy.
Yeah, I mean,
so that involves a lot of microfluidics which I don't know. Okay. So that is a question for microfluidics guy. But I'll tell you, there are small chambers. For example, different companies come up with their different protocols. So the two of them that I know of, one is the fluid ion one. So there there they have the small bulb-like structures where the cells get stuck, and you kind of start from there. And there is a new technology now where they bomb kind of bubbles, and those bubbles actually contain individual cells. And there is one big problem in single-cell technologies is that, and everything is done within the bubble. It's beautiful. It's like within a bubble you have a cell, and that cell you are doing everything. All the chemistry you want to do with the cell is done within the bubble, and that is very cheap. You can see the amount of material you are spending for this cell is very optimal, isn't it? Whereas whereas in the previous kind of technologies which were existing, they used to spend a lot of reagents unnecessarily, which is not true for this. This is called dropsy. Now, one big problem with this technology is that you have doublets, right? So, what is the guarantee that what you are detecting is actually a single cell? Because that's our assumption. That is the kind of axiom that you start with, right? But that is not true. We have seen that in the most well-known single-cell technologies, the rate of getting doublets is as high as 20 to 30%. So now people are trying to get rid of that particular limitation, but I think that's not easy, and it's not at all easy to detect doublets informatically as well.
Another problem to add to this is biologically identifying that cells. What you said is technique-wise how to isolate that cell. Actually, I was trying to ask how to find that this particular cell is going to be a major cause for the cancer or whatever we are studying.
Oh, you were talking about the informatic aspect of it, right? You're talking about once you have the single cell libraries, then how will I know that this particular cell is causal for something or
something like that?
Yeah, that will come to later on.
Millions of cells.
Yeah, we have just started. So I think we have a lot of slides describing how you can use informatics very nicely to get all those cool answers. Okay. Yeah.
Cells not function in solitude means there is a single cell. For example,
Correct.
studying Alzheimer's disease.
Yes.
I know there are neurons, I know there are glial cells and different kinds of brain cells, and they interact with each other in function. They do not function in solitude. So even if I take a single cell and do all this exercise, will it give me a relevant biology answer?
It will, and it will not. Where it will, for example, this is a brilliant question, right? And it is not a brilliant question as well. Because in Alzheimer's, this question really matters. So what she is saying is that if you have a single cell, that is actually sitting in silos, right? It is not connected with the rest of the cells around it. And that is a problem for studying Alzheimer's, right? Because in Alzheimer's, everything happens through interconnections. I don't know about it, but everything happens through the signaling that goes on within cells of different types. And you cannot really study a cell in silo. That's true. But, for example, that may not answer that question. But try to think about what questions we are trying to answer with this kind of test. We are trying to answer, number one, diversity of the transcriptomic diversity. For example, if you talk about single-cell RNA-seq, right? We're talking about transcript because I understand the question that you are asking for Alzheimer's relates more to RNA-seq than DNA-seq, right?
Suppose if I have to see,
How does it matter to DNA-seq?
for example, if I have something like, I want to check inheritance patterns in Alzheimer's, I have some mutations. I do not know where in brain they are sitting, whether they are in glial cells or neuron cells or astrocytes, I do not know. So how will I pick up those genes? For example, I'm just looking at one gene which is
Wait. So how is it more inconvenient than the bulk assays that you have been doing? You can sort your cells using the fax machine, and you can still get the single cells of particular types, right?
Yes. Right now in practices we take blood. We take blood.
Yes.
And since, in my case, if I'm studying inheritance patterns, I know they are in the germ cells. So they anyway will be in any cell that I take. That is provided if I have to take inheritance pattern information. If I don't take inheritance, there are sporadic cases of Alzheimer's. And I do not know which cell on the brain I have to consider for this kind of mistakes. But I know
Correct.
that this is it. How will I go
Oh, correct. Okay. So there is one answer to your question basically. I think what you can do is combine DNA and RNA-seq of single cells. Okay. So what you can do is you can do these things separately, and you can do these things in a combined way. For a single cell, you can amplify both the DNA and the RNA. It is possible. And there are at least by now there are 3 very who is doing that? Snipping. Okay. So there are at least 3 very high-profile papers which are published, which explains how to do combined DNA and RNA-seq of single cells. So what you can do is, once you have the RNA-seq data, you can quantify the gene expression, right? And once you quantify the gene expression, you can actually do lineage mapping, right? And once you do the lineage mapping, you actually know that, okay, this kind of cell, for example, I have worked with single brain cells, and it's not really hard to map to their respective lineages, which are already known. But then also you will miss out the ones which are not known. But finally, you can also consider them as an outlier or a different cluster, very tiny cluster of linear cells or something like that. And then you also have the DNAs. So you have the whole answer, actually. Right? Isn't it? You will have the whole answer. But I thought your initial question was, I answered your initial question, right? We are not really, we are not learning about how the signaling works, but that too can be done in principle. I'll tell you how to do it, but not right now. Yes, please.
Any kind of analysis in which we are trying to analyze a sample from a tissue, and tissue is composed of 2 or 3 different deposits, as you shared.
Right.
So in any such case, one has to go through all the cell types. Single cell RNA for all the cell types. You can't predict anything by doing RNA but for just one cell type, as you shared. Because in our case, there are more than 2 or 3 cell types who can work in a network and all. So we have to have RNA data for all the cell types actually. Single cell RNA data from all the cell types.
Well, that's not exactly true, because you can either have, for example, cancer. Okay, you don't know all the cell types, but for example, you take some tissue from lung. Not all cell types you know here. You can do FACS sorting as well, and you can still do. For example, you want to study the 81 cell from lung. Okay, so you can do a FACS sorting, and you can still do single cells because you don't know what is happening downstream. You don't know what is the heterogeneity. Even if you take 81 cells, you'll find that they have their, they have different subtypes, right? And you can, one approach is that you start with the whole thing, you start with the tissue from anywhere you pick up, let's say blood, okay? You pull out blood, you have 4 different kinds of cells. That's one way. Another way is, okay, I want to just study monocytes. Then I'll fax monocytes and let the single cell.
But in that case, whatever information we are getting is from all the cell types.
Right.
And this will not solve the answer like which cell type is giving signal to which cell type actually.
Which cell type?
Which cell type is giving signal to which cell type?
So signaling, it is not meant for signaling. We are not yet there where using single cell RNA-seq we learn about signaling. But there are many insider things that are being done right now, where you can still keep the tissue in a fixed form and you can still learn about the cells. Yeah, so you'll have to do it. There's a very tedious process.
And then you have the gene expression. Then you know this cell is sitting here, this cell is sitting here, and this. Then you know exactly where they are sitting. Maybe they are same cell types. I did gene expression, and they are sitting at 2 different locations. So we can say that they are giving different things. So nowadays in single cell, what they're doing, they are making 3D maps of tissues. So they actually bookmark the cell locations. This cell is coming from this.
But those are all informatic approach.
Yeah.
So what is done there is basically, so there is a paper, what they did was they took the olfactory lobe and then they did the normal RNA sequencing, and then with some informatic approach they came up with where these cells are, for example, one type of cell is here, one type of cell is here, another type of cell is close by. So those are all are actually based on the assumption that if your gene expression values are similar, then you are close by.
You can also,
So that is the assumption for those kind of things.
you can cross it, that is real issue.
A paper came up in Science very recently,
You can cross
where,
it.
yeah, so that is like, geographical kind of diagnostic taken very recently, I guess 6-7 months ago, there was a paper in Science where you can, I don't remember, so if you have a tissue like this, you can do this all, you can still have the information about their location, and you can do the RNA. They invented a method for that.
So sir,
But that's not for single cells.
sir, if we're going to say a single cell experiment, we have to isolate the cells. Depends on the particular marker. Well, actually, we need to
Not really.
identify the
It
cells.
always doesn't depend on the marker. That is one situation. For example, you were trying to study the heterogeneity of cancer
Yes.
cells in the blood, okay, which are very rare. And you want to study the heterogeneity. So what you do is, because they are very rare, you want to enrich for the cells. Otherwise, in 1 teaspoonful of blood, you will just have 4 to 5 circulating tumor cells, right? So you have no choice other than using EpCAM or such kind of surface markers to enrich for that particular type of cells and do the further downstream things. But that's always not true. For example, you can crush the whole tumor and you can do a single cell sequencing of, let's say, 5,000 cells. And you will learn, that doesn't require any marker-based selection of the cell. It's not always true, but it's true in some cases.
It will be more clear if you give the example of your own paper.
Yeah, I
thought.
So I'm coming to that.
Why you
It's just
choose
that
the
I'm
single?
caught up by so
Yeah.
many questions which I'll have to answer.
Right. So my question is why you choose the single cell sequencing in your case?
In my case?
You said that brain tumor somewhere you did some study.
Oh yeah. Let's come to see some of the examples. Okay. We are going slow. Okay. So let's go a little faster. So here we have this brain tumor. Okay. This is a very seminal paper published in Science. Am I still audible to the rear row? Okay. So we have a brain tumor here. So you are getting the tumor out of the brain, and you are dissociating the cells, and you are catching those cells through your microfluidics into one chamber, or maybe a drop, whichever you want, and then you are doing the further things. Okay? So here you can. So this is a bulk sample. Okay? So don't consider this for the time being. So you are getting single cells from here. This is doing tight, so this is bulk. So you can come from here to here as well. Okay, you can culture it. So what they did was they cultured it, because it is always better to culture the tumor cells. Because otherwise, we have always seen it takes some time to load your cells. When you take the tissue from hospital, it takes 4 or 5 hours to finally load the cells onto your experimental setup. So within that time, a lot of iron degradation happens. So it's a good idea to actually culture your cancer cells, and that's helpful for all other purposes. So what you can do is you can culture that, or you can directly. In fact, we have a paper which is coming in Nature Genomics next week or sometime very soon, where we didn't really do this culture stuff. Okay, so we just generally what we did was we directly sequenced the single cells, and we got the data, we got the matrix, and then we studied about their CNVs, the patterns, their expressions, the clusters of them. So these are something which are normally done. Okay, you get single cell gene expression data, you do cluster. By cluster I mean machine. There is a machine learning term, right? Getting cells, you have a lot of gene expressions. So those are your features based on which you want to learn. Then what are the groups that you can see? And that's the whole point. So the first level work is to cluster your single cells, and then onwards you can do. Because here, one thing you'll have to realize that when you do single cells, that means you want to study heterogeneity. There is no other question. That's the first question we ask. What is the heterogeneity in my data? So what you do is you do clustering of the cells. Once you get the clusters, then you study further and you characterize what these clusters are and so on. Right? So that's the workflow of single cell study. And here, there is a very nice picture. Okay? These guys were very intelligent. They're from Harvard. And what they did was, and I later on realized that this is a wrong figure, but nevertheless, it's very interesting. So these are your genes, okay, and these are your chromosomal axis. So think about a pseudochromosome, okay? Because in DNA, so what is the proof? In cancer, there is a very big question that you will have to still answer, even after you do the single cell. Is what is the guarantee that the cells that you have taken are all cancer cells? Right? It's not very easy to guarantee that. Because in single cell, you cannot really play around with it. You try to prove that, oh, I have this mutation, this is a cancer cell, and I still have a lot of that tissue, I want to do the RNA-seq and all. It's not like that. You have a single cell, you cannot mess around with it. Okay? You'll have to have it intact. And now, what happens is, because you have these cells, you don't know whether they're cancerous or not. So what they discovered a very nice informatic approach. What they did was, they tried to show the CNV pattern based on gene expression. That's very uncommon. That's very new. That's not very new. There were 2-3 papers earlier. But this is exciting, isn't it? Because see, for normal cells, so these are your normal cells, where pretty much everything is, these are your normal cells. So these are cells, okay? So these are all cells, and these are your gene expression. Okay? What's that? It's not nice to write it here. So let me write it here. This axis, you have genes. And in this axis, you have cells. Right? Yeah. So basically, you are trying to, what is this? A pointer, I have not very good. So basically, here, you have the genes, and you are trying to see that, you are considering that, if you let's say this is your chromosome, right? And you sort all your genes in terms of their geographical location on the chromosome, right? That's what they're doing. And you're somehow showing an average of the expression, a continuous average, a moving average of the expression, if I speak more mathematically. And then what you get is a signal of whether this part of the chromosome is actually amplified, or whether there is an extra copy there. Right? So, for example, this brown areas are actually places where they had few more copies than one. Right? Whereas here you have a damp, right? You have lost that place in the chromosome. Right? So this somehow shows you even the CNV pattern. This is a pseudo CNV pattern which you can obtain through simple informatic analysis. And believe me, this is not very tough to do. If you look at the algorithm, the informatic guys may find it really interesting that just from gene expression, you are getting a sense of how their genome looks like, right? How their DNA looks like, and whether there are multiple copies or less copy or one. And that actually tells you whether these cells are cancer cells or not, right? For example, in these cells, you can see there is no such weird patterns. They're all white. Whereas in these cells, you have some patterns. Not only that, what is more interesting is that you have multiple kinds of patterns, okay? You have different kinds of patterns, at least 9-10 different patterns that you can observe from this data. And these are actually one individual patient. So this box corresponds to one patient. This box corresponds to one patient. This rectangle corresponds to one patient, and so on. So you have so many cells of this guy. So many cells of this guy. And you can see that all his cells do not look alike. So for this guy, the cells look more or less alike. But for this guy, look, here you have CNV, which is not there here. Okay, so it's pretty obvious. At least you can see, you can figure out, if you look at this picture very carefully, you can figure out there are 4 or 5 different types of cells, even if you look at it in terms of their clonal, identity, right? Their DNA. And then also you can do, so this paper is very iconic. Although a lot of things here are pretty wrong. That you will discover once you go through this paper and analyze this data yourself, right? That I did. That's why I can tell you that this is not genius. No, so what happens is you have a chromosome, right? And your genes are being transcribed from, so you have a DNA, let's say, right? And all your genes are getting transcribed from particular locations of DNA, right? So if you just organize the genes in terms of how they're located on the DNA molecule, then you can get a sense of whether if the genes which are in this particular region are amplified, that means possibly there is a CNP, there are extra copies, right? So that is the kind of inference that you are doing. And that actually tells you that, oh, looks like these cells have a CNV, so they are cancer cells. That is one kind of, this is an informatic proof of the fact that you are working with cancer cells.
Is there any correlation in these samples in terms of if they are same kind of tumors or if they are in the same?
You guys, tone down your voice. Thank you. Yeah.
So are they from the same stage of the cancer or?
Well, these patients are actually all treatment-naive patients again. So they have not been treated. They are just diagnosed with cancer, and they have different stages as well. So the stage information is also there, and that also has some correlation with what kind of cell types you grow. So basically, if you analyze this data, this is just a picture of how the DNA looks like. But you can also do clustering of the single cells just based on gene expression and the following things. Then you can see that the type of genes which are being expressed, based on that you can get different kinds of survival, and all these things are there. I mean, that also sometimes correlate with the stage of the cancer and so on. That is a big study, so there are a lot of results in the paper. I would recommend you to go through this, Patel, and as an Indian guy, to be proud, and then Science 2014. This is the first cancer single cell paper which came out. And after that, there are very few papers. I think we have one of our papers in the room. This is the next application. So that is, the previous one was just an application of single cells to study the heterogeneity. And the heterogeneity was explained in terms of clone. Right? So that was clonal heterogeneity in true sense. And you can see within the same type of tumor cells from the same patient, you can have different subtypes of the cells. That is spectacular. And based on different subtypes, when you have different subtypes of some kind of cells, you can see how the survival correlates with that. The survival analysis is another statistical field which has a lot of challenges from single cell field right now. So there are many beautiful problems, informatic problems which can be designed. You guys can talk about this later on, but let's first cover this scope of this talk first. So this is another application which is just possible when you have single cells, which is not possible without single cells. Okay? Why? Because first of all, what is this? So when you have a lot of cells, and the cells actually represent different time points, right? So for example, one cell, a cancer stem cell, if I talk in cancer terms, you have cancer stem cells, and then you go downstream, you have other kinds of heterogeneity, and then you have epithelial mesenchymal transition going on. So some cells are actually outbreaking their existing primary tumor and reaching out to different organs and so on. So these cells actually, the cell, you start with one cell, and these cells eventually evolve, right, to divide. So the cells are actually snapshots of one particular time point, the evolution process. So you can think of cells when you have the gene expression or when you have the DNA. They actually, so I'm talking more about gene expression. The reason is that it is, it has wider applications. So what happens is that each single cell actually relates to a historical event, right? A particular environment, something which happened earlier. So it is more or less like lineage tracing, right? So we call it, so we are using a fashionable term for it, which is zero time. Okay? So why zero time? You really don't know this is the time. This is the time axis. So we are trying to somehow mimic what might have happened with the cells that we're studying. Right? This is something which you can do only when you have single cells. Because this kind of resolution, so many cells you don't get from bulk studies. So many samples you cannot obtain. And from bulk studies, what you get is just the average, right? So it's not possible. So this algorithm is very interesting. I'll try and describe the algorithm later on after a few slides. Just the steps of it, not into details. But this is how the zero time looks like. Okay, you're starting all the way from this cell, you're coming along this path, and you're reaching here. So this is no, there is no branching here. So this is a bit not so interesting thing. But there are cases, if you use this software, this is called Monocule. This is published in Nature Biotech. This publication is in Science, which uses that software. But what it does is, it also gives you branches of the tree. For example, you can think of this as a branch, which is not big enough. It's a twig. So then you have a branched kind of a structure which gives you a sense of how the expression, how in terms of gene expression the cells have evolved, right? So, and why we do that? Okay, that's the biggest question. So let's say we can find out the zero time, a hypothetical time axis for your cells. Now, how does it help? Because look, if you sort, then you know that there is a particular kind of ordering of your cells, isn't it? Once you know that my cells are ordering, then you can look back at your genes or your transcription factors, and you can see that how they're shooting up or down, right? At what particular phase they're more active, and let's say how cells evolve, right? This question is very important from developmental angle, right? Developmental biologists are really very keen to understand this zero time and how this transcription factors are changing their expression and what point they're triggering up and what point they're down. So you precisely getting into the, this is the level of precision that you can get from single cells. You know, at this point, there was a historical event when this was up, and that probably caused a lot of cascading impacts on the cells which came out of it. So those kind of analysis you can do, and if you sort, so in this figure, what basically this shows is following. So these are your different genes, which are split across different clusters. So you can do gene clustering. It's not a big deal. Everybody can do it. Right? So you can do a gene clustering, and you get 1 through 11 clusters. And what you see here, these are all your single cells. Okay, and they are organized by the zero time. They're organized. This ordering along this x-axis is according to the zero time. And once you order them and do a little bit of smoothing, it's just like rubbing the expression in this direction. Okay? Taking a moving average, which is taking a trend along a certain short stretch of span. Okay? Here's few cells. So what you do is you take a moving average of the expression after ordering the cells in this way, and you get this kind of very beautiful figure, which you never saw before, right? And this tells you how the transition happens. It's a continuous process. So single cells actually help us to appreciate the fact that all biological processes are continuous. So this is an analysis of the continuum of a very specific biological process. So these are the cells from, I guess, olfactory lobe. And and that relates to how we sense the smell and all. So this came up in Science recently, in 2015, not recently enough. But that gave them spectacular insights into how we really sense smell. Okay, and very interesting study. Let's look at some other applications too. Rare cells. this is another thing which is not possible without single cells, right? How do you get the rare cells? Just had you taken the average gene expression of this bunch of cells, then you would have lost this signal, this very intriguing signal. This is a different cell type. So what they did was they did clustering of the cells in the gene expression space. And here they're basically showing you a visualization of the data. And they got basically two clusters. One is this red one, another is this yellow one. And this red cluster happened to be one particular kind of cell. So these are like 2.5 thousand mouse embryonic stem cells. So this is actually obtained from dropseq data. I was speaking about doing library preparation within drops. So this data comes from that technology. And right now the biggest single cell data that we have is 68,000. Can you imagine? One particular kind of cells, let's say 68,000 blood cells. Now think about what are the things that you can do with that amount of cells. You can do wonders. You can find new cell types, you can find heterogeneity in cell states, cell types, and whatnot. We'll see glimpse of the dropseq here. That's very interesting. That's more new. Okay, they have started getting venture funding right now. There's a company called Genomics, which was just launched, and they have published a paper in Nature Communications, where they floated near about 0.2 million single cell expression, whole transcriptome. Just imagine, 0.2 million whole transcriptome. It would have taken you to do 0.2 million whole transcriptome if had you done it in bulk way, right? It would have taken you like 5 years with all labs working across the globe. So one experiment gives you so many cells. Okay, so this is one rare cell type, and then you can do informatically. You do clustering, you first do dimension reduction. I'll come to what dimension reduction is for the biologists. So, and then they validated that this cell is a very rare species. Okay? So this is one very straightforward application of single cells, which is new cell type discovery. Okay? That's one of the reasons we do single cell assays. So what I do here is that we have somatosensory cortex. Now this is a science paper, came in 2015, and at that point of time it was the paper with largest data size, which is now not true. So you have 3,500 cells from somatosensory cortex and hippocampus, and you were doing single cell sequencing of a lot of cells from these places, and you were trying to find out what are your clusters. Now what this figure is. So you basically have cells along this axis, and you have genes along this axis. Okay? And you are trying to find these boxes. You are trying to find this mosaic pattern. That means certain genes are coherently expressed in a group of cells. That is the meaning of this particular figure. And you can find these boxes. Now the method which was used to find this is by clustering. I think many of my informatic friends will know what biclustering is. That means you want to cluster both genes and cells together concurrently, and that's super slow. I guess some of my friends here, the students working on faster version of biclustering. It will be very nice to see what happens. But I consider this to be a very inefficient method, and this method takes days to give you very nonsense results. So anyway, these are big guys from, I shouldn't say all this because it is webcasted, right? So I realized that every time I'm trying to memorize, I'm forgetting that this is being webcasted. Okay, so I don't know what to say. So sorry to all those bad comments. So this is a brilliant method, and here you have this, I was talking about some other method. And you get this kind of mosaic pattern. And this happens to be, all these are different cell types as you can perceive. Right? And they actually got 47 subclasses, 9 classes. So 9 big boxes, and within them they go even further down, and they try to discover the subset types even further, and they find 47 subclasses. Now that's something very odd. 47 different types of cells. You hardly know some 11, 12 types of cells from brain, right? So this is too much. 47, and they did only provide evidence for 2, 3 different cell types. They did the staining. They proved they really exist. It's a good job. In a single paper, you were showing 2, 3 different types. But again, it's unbelievable. And their informatics was a little questionable too. So here you can see 2 different types of astrocytes here. Okay. So this GFAP, they found through informatics. These are all not known beforehand. Okay. They just know there are astrocytes. But what they found was that within the astrocyte box, let's say this is the astrocyte box, there are other cell types. There are even, the diversity is a lot more than what you find in the box. And what I see is there are 2 types. And one is astrocyte 1. They name it astrocyte 1 and astrocyte 2. Had I been in their shoes, I would have given the cell type name after my name. But they were not innovative. They called this cell type as astrocyte 1 and astrocyte 2, and they came up with different markers of them, and then they showed that they really exist. So that is for the aspirants of single cell biology, what I'd say is that's the most conventional workflow for single cell studies. You sequence cells in large amount, as large as you can afford. Okay, you do very nice informatics. You come up with cell types. You stain, you prove that, and send your paper. That's it. That's the pipeline for single cell studies. Okay, and you can do all these attractive things like zero-time analysis, which gives you understanding of how the factors work, how different transcription factors actually trigger different events. That's also very nice. Now with that, I think we're done with the applications part. Any questions? Any bottle of water? There's none, okay. Okay, so, tell me. Yes. This sense? This sense. No, these are taken from mice. Okay. Mice, directly from the brains. They are not cultured. It's very hard otherwise. So that's why you do all the studies with brains. Normally there is just one study. People have done a lot of single cells in brain. Hey, thank you. I didn't mean that.
Why not?
It's great that you have my friend Ganesh. Thank you. So, to answer your question. So normally, I just speak so bad.
I guess you could continue, we'll have you back.
Okay. So it doesn't stop us from answering the question. So normally, when you try to study brain cells, it's better to do with mice. Because post-mortem brain quality is not that great. Okay? And you have a lot of limitations. And the quality is not great. And there is only one study so far, which is published in PNAS by Steve Quick's group in Stanford. And it's worth taking a look at. So those are all human post-mortem brains. They didn't really discover very new cell types, but because that guy, I like his work a lot. He doesn't overpromise or something. He just came up with he speculated some cell types, but he didn't say, one cell type I think he proved that this is a new cell type. So otherwise, all the studies are actually done with mice. And then you can immediately take the cells. Oh yeah. Yes. So her question is that, have you cultured the cells, right? Or have you taken the cells directly from the tissue, from a living individual, or from post-mortem urine? That's not nice, somebody wants to take some cells from anywhere. I would say no, no way. The mice cannot do that, right? So there is a difference. The cultured cells are much better in quality, right? And you see a lot of genes expressed. The number of dropouts is very less, and it's a lot better to work with. Informatically, it's a lot better to, just note my word, informatically, it's a lot better to work with cultured cells, but not biologically. Because my friend Vibhor has some experience with dealing with cultured cancer cells, and I also have such experience that where we have seen that we invested so much to learn about the heterogeneity for cultured cells from different individuals, but we didn't find heterogeneity. Certain kind of cells actually grew in that environment and all other cells didn't. So you will lose a lot on the front of heterogeneity, which is the sole reason for doing single cell assay. So I won't suggest you to do sequencing of culture cells. Yeah, I won't. Because you see a lot less heterogeneity there. But informatically it's brilliant. I mean, the cell quality is splendid. Okay, no other questions. So we move on to the next part, which is the data analysis part. And here, first word of caution, although single cell is great and all that, I have been saying, but it doesn't come without any expense. The expense is huge. It's not only the money that you spend, but the amount of noise that you buy, right? You buy a lot of noise. And the noise, you don't know when you see a zero in the matrix. You see a lot of zero. I think 70% of your cells in the n-square matrix, not n-square, right? Gene versus gene times the number of cells matrix will be zeros. Now, you don't know whether the zero is for low expression, low expression of the gene, or whether there is a failure of the technology or the chemistry. It's very hard to distinguish these two sources of noise or the dropouts that you get to see in your matrix. So it's a lot, and also the other part is that the cells, although you process them through the same pipeline, at the same time, let's say within the same batch, you see different levels of noise in different cells. You see that in some genes, some cells, the number of genes expressed is around 1,000, some cells is 2,000, 2,500, which is too varied, isn't it? Because in bulk samples, we don't see that kind of variability. The variability is a lot less. So there are many other sources of noise. It's very hard to explain all this within this short span of time. And even if I explain, you won't take it nicely. I think you'll come and hit me. So I would stick to the basic. I'll just hand wave and say that, this is very noisy. And the kind of data that you get are very noisy. And what you need to do is you need to do a lot of library checks. You need to see that whether you have the right kind of depth of the specific cell. You need to see whether you have enough reads. Let's say at least 1 million or 2 million reads are okay. But if you go below that, if you go to 50,000 reads, that's not nice. Right now people are doing that. For example, in Dropseq, when they produce data with Dropseq, the number of cells that they get is very hard to sequence, right? 68,000, 70,000 cells. How much money you can pour in? So they finally sequence for 1 lakh reads, okay? But for other assays where you're not doing that much, you're just using 100 libraries, 100 cells. In those cases, you can still invest money. You can say that, okay, I want 4 million reads per cell and so on. But even then, you'll have to check the library quality. You need to see whether you have enough reads or not. And then all these metrics that you need to do, which normally you do for any bulk assays, right? So you can check that, then you have to, yeah.
Sorry. So you're saying these 100 libraries will give only 4 million of reads?
No. Each library.
Each library will give 400.
Each cell. Library, I don't know if it is a correct word.
And you use
For each
400
cell, you expect 1 to 3 million, 4 million libraries. And that is good enough for informatics. But if you go below that, it's a little questionable. It's, because the quality is varied. That's the problem. Even within the single run, the cell quality are very different. And that you will get to know possibly when you do the hands-on. I'll try to show you how it is different, even within the same pattern.
So getting 100 libraries is big task. You have to spend a lot of money, I think. Right?
You had to spend a lot of money 1 year ago, but right now you have methods by which you can save a lot for reagents. The reagent cost is a lot, right? And also downstream, you'll have to save money for even if you save money for reagents, you'll have to save money for your sequencing too. It's not only reagents, so it depends on how much money you have.
So I never worked on the single cell, so I want to know how many libraries you prefer for a single.
Normally the kind of work that we have done back in Genomics of Singapore, for one individual patient, we used to have one chip of fluidine. Okay? We were using fluidine chips for single cell capture. And we used to have 100 cells out of 96 cells actually out of one plate. So, and now I think they're going to buy this new platform. So Singapore is really a leader in this area, in a lot of areas. So now they're going to buy the drop-safe facility as well. And there I think they're going to do like, already have, for example, the same company, Fluidine, has their chip for 800 cells as well right now. So we did 100 cells when I was there, and now they're doing 800 cells, and now I think it will be increased even further with the advent of the drop-safe. It will shoot up to 4,000, 5,000, 6,000, to 40,000, 60,000. Now it depends on your backgrounds. So once you do the read alignment, so you get reads, you are happy with the quality of the reads that you're getting, and you just, also there are ways of selecting what reads you want to finally hit, right, for your analysis. So that. So there are certain scores, the quality scores. So we just say that, okay, if this is more than 50, we'll consider this, otherwise not. And the quality score is actually a function of the quality of the bases that you're reading. So finally you have this, I don't know what, grade scores? Right. So based on that you decide whether you want to keep this.
It should be more than 30.
It should be more than 30 to 450. It depends on how many reads you have. Okay. Right? So then expression quantification in another task is another task where you use couplings or softwares like that. So because you have aligned all your reads, so you know where those reads belong to in the genome and in the transcriptome, and then you want to, that leads to the quantification, then you know the gene expression. And once you know the gene expression, you can do all these interesting things that you have seen. But to reach this is also a big data problem. What's going on? I've never touched this. Oh, that's it? Okay. So this is a big data problem. Why? Because if you want to quantify the read alignments and all, if you want to do one by one, then you'll spend a lot of time. When you have 60,000 cells, you cannot do one by one, like you did for bulk. You'll have to process all of them together, so many rates concurrently. Now for individual cells, you can assign a different core or something. And there are many methods for sequence alignment. I don't know what methods you guys use. Top hat or something? Top hat? Yeah, so top hat is super slow. So now what you have? That is also super slow. So the new tool is star. Okay? Star is in memory. It occupies 40 gigs of your damn PC. You cannot run it in PC actually. So and that actually saves the whole genome inside the memory, main memory. And that's how it works. So this is very fast, phenomenally fast. And once this is saved in your memory, then it can be actually accessed via different ports. You can centrally store it, and it can still be accessed. So these are all paradigms of computing. It's very interesting. But star is the tool which is more appropriate for single cells. And then finally you use coupling or HDSEC depending on whether you want to have the read counts, whether you want to have the FPCAM. For all that you can use different softwares. If you want FPCAM, then you want couplings. If you want count, I think Vibhor will show you some of those certain things. In the hands-on, you want what? For counts, correct. For counts, you need HDSEC. HDSEC is very fast. So you can either work with counts or work with statisticians who love counts. I don't know how many, any of you statistical genomics here? No, okay. So they have, I know, some kind of bias towards counts which is not understandable.
Coupling also gives count.
Is that?
Couplings also gives count.
Oh, that is not visible. Okay. Coupling count is not the, I'm sorry to say that. Okay. Coupling count comes with places after decimal, right? It's not the count, it is not an integer all the time. If you look at the coupling counts, those are not always integers. You will have real values as well.
All the integers you have a PK value, count, P value.
I know.
Yeah.
Most of there is a column for counts. Most of the values are integers, but some of the values are not. And those who I don't know the background science a lot for that particular field. But what I can tell you is that people who use count, they don't use couplings. Okay, coupling counts. But I have seen people using RACM counts. So RACM has been shown to be a better software than coupling.
Both works on the same principles.
What's that?
Principle-wise.
Principle-wise, they are more or less same.
Right.
But increasingly, that paper was not published in a big journal.
Yeah.
We'll look at RACM paper. That's published in Bioinformatics, BMC Bioinformatics. But that's the fastest growing software, and people are using that frequently. Now, HDSEC I prefer because this DSEC or AJAR for the differential expression analysis, these tools actually prefer this HDSEC count data, which are simple read counts. Okay, so I spent a lot of time on that. So cell filtering, gene filtering, that is, you throw away the cells which have fewer genes expressed. You throw away the genes which are expressed in fewer cells. Now this is a bit confusing. You do both actually. You want to throw out the cells which don't have a lot of genes expressed, and you also want to throw away the genes which don't have a lot of cells where they are expressed, right? So if you do all this, then finally you'll be left with a matrix where you'll have just 6,000 to 7,000 genes, starting from 30 genes, to finally we'll have like 6,000 to 7,000 genes, and for poorer quality data with even worse quality, you'll have genes even lesser in number. Okay? So then you do normalization. That is a very crucial part. Normalization is not yet solved for single cell. For bulk, you have different methods, but how to normalize single cell data is not really very well known. I have a paper on that, but I am not glorifying myself here. So that works better than other methods. I'll give you the link of the paper. Okay, so once you normalize, you have to make sure that what you're learning is actually biology. It's not some kind of weird noise. That's the sole purpose of doing normalization. And you want to level them, right? One is here, one is here. You want to just make them more or less stable. That's why I do normalization. So unsupervised clustering is the first step because you're starting with loads of cells. Okay? So you want to know that what are my types. Because here, the key difference is that for bulk cases, you used to do case versus control kind of studies, isn't it? Because you had very few samples. The technologies were very poor. Now you can do a lot of samples from the same tissue. You want to investigate even further what are the new things. So the reason you do this analysis is to find new cell types, right? That is the main reason, or trying to learn something new. So when you do that, the first thing you'll have to do is unsupervised clustering. Unsupervised clustering is, what is the commonest method for that? Somebody? What is that? K-means, right. Polysemen, nobody uses that. So biologists are simple guys, okay? You give them any piece of crap, they'll buy it. But K-means is one, certainly. Then, what's that?
What is that K-means?
Yeah.
One of my friends actually doing it. I have the papers. One recent modified K-means has come.
Okay, so modified K-means. Okay, so K-means has a faster version as well, which is known as mini batch K-means or something like that. That works very fast, but K-means has a lot of problems. Somebody can tell me what is the problem of K-means?
Local research.
What is that?
You have to specify the
And what else? Quick. I know there are a few machine learning people are hiding here. I know all of them. So can you discover this kind of clusters using K-means? That's a problem. And that is a problem. That is really a problem, and I have seen that not working in single cells. Okay? So,
why does it work with K-means twice?
please attend my machine learning talk.
Okay.
That doesn't work because K-means is meant for finding balls, right? It's kind of, it tries to find, think about this. So this is a safer example. This will never work. This might work, but not sure. This will try to take this part as well. But this will certainly not work. This is, so who can tell me what is the most basic method which works in those cases? What? What's that? EM method. K-means itself is an EM method. What's that? Very good. DB-scan works here. No, okay. So DB-scan actually finds you this kind of clusters very easily. And what is the other method? Hierarchical clustering can do it? Yes or no? Yes. Hierarchical clustering can also do it. And also network-based clustering can do it. All these types of clustering can do it, but not because this came on. So K-means is the godfather kind of thing. It was there ever since. So obviously it is not able to do it. But it's still one of the most used. Okay. So K-means is very fast, very rapid, but it will not detect very important clusters. DB-scan is terrible. You know why? Because we have two parameters there which are very hard to set. And you never know until you visualize in a lower dimensional space, you never know whether the clusters you are getting is actually correct or not. It pretty much depends on how you set the parameters. And those two parameters, it's very tricky to set those two parameters. It's tricky. Okay? Now what else? So we are done with the clustering. Okay? So clustering, the importance of clustering is that you find the groups in the cells. Okay, you have a lot of cells. You find the groups. You have a tiny cluster here, you have to get it. That's important. But even before that, okay, this is gone. Okay. So can you increase the time you work for this or somebody? The time is it can be adjusted before the monitor goes off. Okay. So your first step is clustering, right? Is it the first step? No. because you want to find which genes you want to cluster, right? Although it is written there as clustering, but I want to tell you that is not the first step. Machine learning guys will appreciate that. So the first step is actually finding out what are the key genes, because remember, you have like 7,000 genes. That's not less. Can we think beyond 3 dimensions? Yes, we can. Time. Can you think about 4 dimensions, beyond 4 dimensions? No, we really cannot think. I think some topologists can think of that, but I don't. I think people can think about, one kid can think about 7 dimensions. That's what his parents claim. So I saw somewhere, it's very funny. But you can just imagine fewer dimensions, right? Our mind is not made that way. So think about when you have so many dimensions, then what you learn, the clustering method finds it really tough to get the right clusters out of that huge dimensional space, right? 7,000 dimensions, just imagine. You're learning something from 7,000 dimensions. Now that's not easy. So you want to reduce that, and reduction also involves a lot of important things. So feature selection, the favorite topic of Indian machine learning specialists. You'll find loads of feature selection papers. So feature selection actually solves that problem. Feature selection has actually 2 parts. One is selection, one is extraction. Right? So feature selection, okay, I have 7,000 genes. I take 5,000 out of it, or I take 2,000 out of it, or I take 20 out of it. That's selection. You take the genes, you don't kill them, you take them as it is. But what is feature extraction? A lot of people have trouble in. So I am actually covering a few things from the later part of the slides as well. So let's think about let's think about a hypothetical situation. Some people you like just like that. Some people you don't like. You don't really need them. Have you realized that? But it's not that simple. Have you really thought the process underlies that? We don't like to stay in company of some guys. I mean, it's love to stay with some other kinds of people, some special. So these are 2. So if I draw this mathematically, right? You have 2 clusters. Some people you love to stay with, some people you don't. But is that, can you describe precisely why you love to stay with somebody and why you don't like to be in presence of someone else? It's not that easy. It's not that easy. It's a very rigorous process. There are so many factors there. And what I'm saying is all these factors are, let's say, your genes. All these factors that influence the final decision whether you like somebody or not is actually a cocktail of so many other things, right? All these factors actually influence the decision. But finally, what you have is, so my sacred thread actually hitting my hand paralyzed. Okay. So when you have multiple such dimensions, so although this may not look like dimensions, so these are so somebody borrowed money from me. Money. Never get back. Right? That could be one dimension. And that can be book. Yes. Book is another dimension. Smell. Some people smell weird. Okay? So that could be another dimension. Right? So all these dimensions, you never know. You never realize that, thought precisely about what is it that keeps me away from this guy. But finally, when you have all this information, you'll see that the best thing that you can do is you will naturally have 2 clusters. And those clusters, so that is feature abstraction. Remember this term. When you do PCA, right? Let's say these are your gene 1 and gene 2. Right? And you want to have this kind of data, you have these points, right? By drawing this back. So when you have this kind of points, and you want to say that, the question is, the feature selection says, Then you take gene 1 and gene 2. That is feature selection. Maybe gene 1 is important. But feature extraction means you want to have an abstract feature. Here, in my hypothetical situation, the abstract feature is whether I like or don't. Right? And I don't have a definition, a precise definition for that angle. Right? That's something new. That is a function of so many different factors. That's exactly what is feature extraction. Right? And you get something, you get a new axis system. This is a new axis system. Which are your principal component 1 and 2, let's say. And these are describing the heterogeneity. So basically, so you'll see the maximum variability of the data. The maximum variability. So this is also maximum variability. I don't like, two opposite poles. So maximum variability is actually along this axis. But had you looked at these clusters from this side, they look all same. They look all human when they are far away, if I don't know them. But as you get to know them, then you try to distinguish between them. And that distinguishing capacity is something inherent, something that we have inherently. And we always do PCA in our mind. We always try to find the abstract dimension which actually distinguishes data that we have very, very efficiently. And that basically makes the things very easy. Once you have 2 or 3 dimensions, let's say instead of gene 1, gene 2, you could have a lot more dimensions, right? But you can still have 2 such abstract dimensions out of it, which actually explain all your data. All the heterogeneity, all the diversity, everything in your data. Isn't it good? Isn't it much easier for your downstream analysis to work on that? So that's exactly what we do with feature extraction. Like the methods PCA and TSNE. PCA stands for Principal Component Analysis. TSNE is another method which is used a lot nowadays, which is called T-distributed stochastic neighborhood embedding. Don't try to memorize. Okay. So this method also is a superior version. I don't really believe that, but it is used more nowadays. But unsupervised clustering. And followed by the dimensional reduction story that I said. You want to reduce the dimension? Question? No. Okay, differential gene expression analysis, that we all know, I guess. What is it? So you have 2 groups. Now by clustering you have got in 2 groups. You want to compare what are the genes which are differentially expressed. That's not an easy task either. That's also a very tough thing to do. Because you don't know. For doing that statistically accurately, you need to know the distribution of the model of the gene expression, which you don't know all the time. For single cell at least, we don't know. So since we don't have a lot of statisticians here, I'll just glide through that. That's one of my favorite topics. But serotype analysis, you have already seen how you can get the nice serotype lineages, right? You remember that plot. Go-term analysis, again, is a boring, poor, old thing, age-old thing. That's very efficient. You can find out when you have a group, by looking at the pattern, average pattern of the gene expression, you can relate the gene expression to the known biological stories, right? So that's what gene ontology is all about. You have to tell a biological story out of your gene expression. And the biological story you can tell from the textbooks, which are actually the GO terms. And then you have lineage tracing. That is the key thing in single cells. And that is the thing where informaticians can contribute a lot. Given a lot of single cells, how do you know that what kind of cells you are getting? You know, so previously, in last many years, we have spent enormous amount of time, money and effort in sequencing all different cell types, right, which are apparently clearly findable. And we have all those data publicly available. Now, given your single cell data, can you map to them and try to identify? For example, her question. When you have a cohort of single cells taken from the brain, how do you know that what cell is glial, what cell is neuron, what cell is astrocyte? Right? So your first level question will be, okay, I have 500 cells. Now what I do with that? I need to know that what kind of cell that is. What known cell type that is. That is the first question that you ask. And that relates to the lineage question. Right? So you have all these things where big data, informatics come into play. Now these are the methods. I'll not spend a lot of time here. Who has a question here? Looks like, yeah?
So which part of your data analysis really had that big data issue?
We'll have. We still have one particular
So
case.
from last slide,
Yeah.
you just which part of this whole analysis has that big data issue?
The big data issue starts from the beginning. From the library, when you sequence reads, right? What I said, I was the only time in the lecture I was talking in terms of numbers was millions, billions, and all these things. Right? So you have millions of reads, and when you want to align those reads to a known sequence, right? You have tiny reads, which you'll have to map to known sequence, which is DNA. That's a very complex problem that you are doing for so many rates is a big data problem, for which you have some solutions already
How much
existing.
data is really a big data?
I should say large data.
Means
It
large
is not
how
big
much?
data. I don't know.
Is it any threshold?
I don't know. I don't have an estimate. Big, how big? I don't know. And big data, the word itself is a word that we like. There is no such definition. It's just like you were talking in terms of terabytes, petabytes, right? So obviously this, when you store the single cell data for a couple of studies, it takes terabytes of memory, right? So the number of reads that you have, so many, it takes a lot of space.
But it is actually the complexity of the data or the size of the data is the
The size of the data is big, the complexity of the problem of the read alignment. Did you study dynamic programming or something like that?
Sorry?
Dynamic programming? You're in Compile?
Yeah, computer.
Okay, so when you have a stretch of DNA and you want to map small fragments, right, to what part, it is a complex problem because not always this, so there are small differences. It sits here, that's fine, but it has certain differences. So what is the optimal place for this? Where do you really, will you place it here or will you place it here? Because there are certain differences. So what optimizes the allocation is a big data problem. And it's for a massive amount of data. So read alignment is one big data problem. Sure.
I'm not getting the answer. Like the complexity. Suppose I'm just sequencing one chromosome. Okay. So I got some reads. Now I am aligning with the whole genome. Okay.
You are not sequencing chromosomes.
Sorry, means one stretch
Let's say you are
of
sequencing RNAs.
yeah. So one stretch of data, and I'm aligning to the genome. So again, I'm having the same type of algorithm using. I'm using the same algorithm. I'm having the same calculations. So means what
The contribution is the algorithm.
yeah. So means does it means like the size of data is the actual
No,
causing the
size
problem?
of the data
or
is the
or
issue. Size of the data is the issue. That is also big data angle, but it's also the complexity of the problem itself. But the complexity of the problem was solved even for smaller amount of data. So I think, yeah, so you can say this is the, for bigger data, when you have single cells, you have like the sample size is much more, right? Thousands. So then even the previously known algorithms are failing. That's why people are coming with new alternatives for read alignment. Because even if you know how to align rates very accurately, you will have to scale it for thousands of cells, which is not easy. So that's the big data angle. I'm just going a bit fast now. I'll catch me later on. Okay?
I think the data size is directly proportional to the big data or something. Because complexity you can get in
Data
small data.
size, dimensionality, everything.
So dimensions you can get in small data. That you cannot say that it's big data. Right? Means
Dimension, sorry.
what you're saying the complexity, what he's trying to ask. Like in the small data set also we can get all the complexity. But that is not a big data for me. If I have to define a big data, I'll say that the size of the data should be big. Then
What
only-
should be the threshold? My question
Threshold
was
is up to
Now, I think what he is trying to ask is, at what part, whether the contribution is to the size, whether the challenge is the size, or the challenge is the complexity.
because now
I would
we
say
have
it is
supercomputers, so I don't think size is the problem.
No, size is not the problem. Means you can store the data doesn't mean you are done. You are processing monstrous amount of data concurrently. Let's say single cell, you have 60,000 single cells, and you have to align each of their rates, which is near about 1 million. It's a big data problem. This is a big data problem. And you need to know how to manage that. Normally we use multi-core systems and parallel computers. I have used up to 2,000 different cores to map it. And further down, I'll come up with one example where I will share with you a little big data aspect. But let me just, I have a few more slides. I think let's talk afterwards. So, dimension reduction, most well-known methods are PCA, reasoning, and representation learning for the guys who are interested in neural networks, deep learning. Although representation learning is kind of slow sometimes, so we rest on the other two. But even PCA doesn't work with very big data. Can you tell me why? What is the complexity? PCA? PCA works on what? What matrix? Yes, a covariance matrix depends on number of genes, right? So if your number of genes is 7,000 versus 7,000, I try to run it in computer, it will not run. Right? So it's hard. And also, not only, so I'm wrong, sorry, what I'm saying is wrong. It's not only variance covariance matrix, there's also the number of cells. But you have to do all those computations, right? That also matters. So I think both the number of cells have, because had it been just the number of genes, it is not very different from the bulk acid, right? Since you have a lot of cells, that adds to the computations, the small computations that you do as part of the PCA. So I have normally seen it doesn't work for more than 2,000, 3,000 single cells if they have like 7,000 genes. Now what to do? Now for all that, we have faced all these challenges, we have tried to come up with new methods. I'll just show you a glimpse of, I think this part is already covered through my discussion. Just to give you a brief idea about how the analysis is undertaken. And this is an example of dimension reduction. So this is one of my data that I used for differential expression analysis. And there you can see all these different types of brain cells. And those are pretty much separated in the PCA space. Very nicely you can detect all the cell types. And this is the big data problem, okay, that I have been talking about so far. So dropseq gives you 60, this is the largest known single cell data that we have. 68,000 single cells are captured in this figure. 68,000 single cells with near about 10,000 or 7 to 10,000 genes well expressed. Now how to deal with this data? So what I have found is that the existing solutions, for example, if you try to run k-means, it will run, but the quality of result is very bad. So the big data solutions for clustering that exist there were not really decent on this particular data set, because we can claim whatever you want, but if you don't really deal with the biological data, you will never understand the complexity and the challenges. So here the data is very nice because we know the ground truth. So what they have, let me tell you a bit about I'll just take another 5 minutes. Okay. So what they do is they fact sort single cells or blood cells of different types, first in monocytes, then dendritic cells, and so on and so forth. And they did facts for 11 major cell types. They sequenced them, they did single cell sequencing of them, and then they annotated each of these 68,000 cells by looking at the average expression of the known cell types. So in some sense, this data is annotated at a large level. But look at the figure. Does it look very nice? No. Because all this, I don't know which algorithm. So they actually, since they started with 11 cell types, they forced the k-means to find out 11 clusters. And they found 11 clusters. Now you see, you get this kind of, these are some known cell types. Color are known cell types, okay? And locations you can see yourself. So these are known cell types. So these cells are getting into these cells and so on. Here you have little pick here. So, and also the signal to noise ratio is poor. And also the clusters are not well separated. The clusters are kind of creating a mesh. That's not nice. That's not a very nice example. That's not a very nice pitch for using your clustering algorithms. So what we do, what I did, and my PhD student, Devachoti, back in Indian Statistical Institute, did was that we used LSH, which is a very well-known big data technique. Okay, so I know. So let me tell you what is LSH. You know, it will be very unfair if I don't tell you anything about big data. So this is basically a picture of few points. Okay, and you try to draw this random hyperplanes. So we are showing here just two axes, but usually you have not many axes than two axes, right? So you just create this random hyperplanes. You are drawing the straight lines, and what you are saying is if 2 points are within, and you are assigning them certain codes. So for example, if this guy belongs to this side of the green line, you will give it a code. Let's say equal to 0 for that bit, right? So you have like 3 different bits here, 1, 2, 3. Okay, this is 1, this is 1, this is 1. And for each of these bits, you assign 0 or 1 based on which side of the line it belongs to. And you do that, you'll immediately see the benefit of doing so. Because you have for those points which are enclosed by the same boundary, have the same hash code, essentially. You do it multiple times, this is a very efficient method, this takes minutes. And you get hash codes, and based on hash codes you can decide whether this is called locality sensitive hashing, where you preserve the structure of the data in your hash code. It's a big data technology which is discovered by a brilliant scientist who is no more with us. Who is that? Who is that? Rajiv Muswan from Kashmir. He was in Stanford. He was a brilliant professor, and he was the mentor for Google founders. Right, and he died in a swimming pool very unluckily in his Kashmir villa. So we used that technique, and we got this kind of a result. Okay, you see the separability of the clusters is spectacular compared to the previous video. And it's very fast. The previous one, which is the current best practice method, takes around 1.5 hours to give you the result. Whereas our method takes 10 minutes to produce this. Only we have come up with the new strategy for dimension reduction, which is very, very efficient, unlike DCA. Okay, the normal DCA. So that gives you, these are the true colors, the annotated colors, and these are the true colors. There you see a bit of a mess, and these are the cluster colors. So when you look at this figure, you kind of believe that possibly this is wrong. Possibly this blue is not part of this brown. But in the other figure previously shown, it's not really easy to comment on the sanity of the method or the data for that matter. So such kind of applications are underway and will communicate very soon. And this is a very fast method for differential expression analysis, non-parametric method. Since we have, if you understand the basic statistics, you'll appreciate that if you want to model something, right, you need a lot of samples, isn't it? If you want to model the gene expression, you need a lot of samples. You want to do it accurately. So now you have a lot of samples, right? So you can use non-parametric method or parametric method, it doesn't matter. But what happens with parametric method is that it's not yet known what actually fits accurately on the gene expression data. So if I get one more minute, then I must say that, okay, so what is the marginal expression of a gene, okay? So let's say this is a gene in the matrix, G1. And that is there across many sets. And if you do the distribution, it sometimes looks like this, it looks like that. But for bulk, they all look similar. So that is something very distinct in single cells. The distribution, the marginal distribution of gene expression is very stable for bulk-based assays if you're doing it in the same types of cells, which is very different, even the same cell type, so very different distribution. And you will call it biology? No. I don't think it's biology, it's pure noise. So how to find the model? So can you find a parametric model for this? Well, if you can find nothing like that. That is my active field of research. Okay? If you can find a parametric model for so diverse family of distributions. But if you can, then the better way to go is non-parametric. But you don't have any assumptions about this statistical distribution of the gene expression. And then we came up with this method which outperformed the existing by a good margin. The work was done with my mentor in postdoc, great scientist Shyam Prabhakar. And you can see the performance is much higher. The AUC values, when you compare with the bulk with the differential expression calls of two different types, we see the match is quite high. For the black one represents our method which is called nodes, and it's much faster for the number of cells here. So as the cell number grows, you can see this methods are becoming they're not working at all. They exhaust the memory, or something happens. They simply cannot scale that high. Whereas these two methods, one of which is mine, another is the bulk-based method. That also works, but that produces crappy result. But this is meant for, my method is meant for single cell, and for obvious reasons it performs better and it scales high. Okay, scales easily. So that's a way to go. That is also a big data problem, because when you have, but big data problem doesn't always mean that you have to solve it algorithmically. You can solve it statistically as well. So this is an instance where you are solving a big data problem where you have higher number of samples. Now we will say that, okay, are you saying 200 cells is a large big data? No. But it is a relatively bigger data than what was used before, right? 200 sample was not used before. And now since we have that in new methods and we came up with, that works faster and better. And this is the zero time I have already described, I think, and I have not only I have, it's just that we have few PhD positions in our lab. In my lab I have 2-3 positions which are funded, and in Vibhor's lab as well there are positions. So we are actively looking out for intelligent people, and not so intelligent that will go very soon. Moderately intelligent people to join us. Okay? And I think my friend Ganesh will spend a couple of minutes. I'm really sorry for taking a little more time than allotted for me. I was actually saying to the organizer that I'll finish well before the time, half an hour before the time. I took 10 minutes past the actual time. So we have two senior friends from our department, Sriram and Ganesh, who will give you an introduction to our department and the kind of activities we are doing here and the kind of positions we have in the near future. And if you're interested, please drop by, please catch us anyone. With that, bye.
So I'm Sriram. I also belong to the computational biology group at KPIT. And I'm just here to just tell you, I'm just going to take a couple of minutes to tell you about the work that which I'm doing. And it's sort of an advertisement from my group. My group is very small, mine and my PhD student. And my PhD student, I'm not finding him anywhere. Possibly he's in the lab. Okay.
That's better.
The type of work that which I am doing is basically the dynamical systems applied to biological problems. Particularly, I work on bistable systems, oscillatory systems, because most of the biological systems are highly dynamical in nature. And so I applied the dynamical systems theory to it, particularly what is said like I built the models from the scratch based on certain hypotheses I built about the biological problems. And I built the models and I analyzed the models. Then I make certain predictions about the model and see that like whether these predictions can be taken to the laboratory and predicted. Okay? So I do not have any big collaborations, but I talk to a lot of other people in the industry as well as in the laboratory like IGIV, and we are willing to do some experiments based on the model results that which I get. Okay? So that is my small group actually. So I am also looking for PhD students who is interested in basically more of mathematics, not something like a big data here. It's the smallest data I use basically. For example, the data that which I get is basically are from the biochemical techniques, not from the single cell or the microarray or the omics data that which I'm talking about. Because there are two ends basically. One side is the big data where you do have enormous amount of data, as you have just now heard. The other side is that from the biochemical techniques you just generate very little amount of data. For example, the time series data for example, if you take a circadian rhythm, it is a 24 hours oscillations where you get data only for something like 2 days. Sampled of every 1 hour, you get around 48 data points, not more than that. Okay? So analyzing the data point, it's also pretty challenging actually. And people who are interested in building some differential equation models which are highly non-linear, who is interested in analyzing, basically estimating the parameters for the models, and those who are interested in working on any biological problems which is amenable to most of the dynamical system tools that I work on. So you can, again my name is Srinath, so you can just go to my website, go to the IIIT website and you can see my name there. If anybody is interested, please send me an email, shoot an email. So time lag of around 48 hours. Okay, so usually that's because I see the email, but I also read the resumes and also the motivation to find out like why you want to work with me. So I take time to just look into it and see that whether it is also suitable actually. But I'm very open to what to say, that I read the resume and see that if you are interested, certainly I would like to contact you and also know more about like why you are interested in the work and what you want to do, and we can have a discussion. So I'll just leave it to you. Thanks.
Thank you. Okay guys. Okay, we hope that you have a great session in terms of learning, and we will be going on forward for the lab session soon after this. Before that, after we break out from here, we will have a quick photo session. So all of you are requested to join us near the lawns nearby, where we'll have a photograph after that lunch, and then we'll be moving to the library block for the laboratory sessions, hands-on sessions. Okay. So quickly, brief information about the positions available with me. I'm in the middle of completing a few PhD students, 3 of them, and we would have 2 positions available in my lab, of which 1 will be funded. The other, one expects that some of you who might have CSI, GBT, etc. funds may want to join. Summer internships starting from May, July. This advertisement for the PhD positions will be coming up in the month of May, June. So those who are interested and those whose friends, etc. might be interested, please inform them. Summer internships, there are 4 of them. All of them are stipend supported in the month of May, June, July. And the requirements are you should be either a UG or a PG student of coming from BTech, MTech, MSc stream, and from various backgrounds depending on the problem. So you will be suggested a problem, and the selection will be based on what kind of expertise that you have, right? And in addition to that, there are running projects which will be for thesis projects on questions of which are going on aligned with the research that is happening in my lab. I would like to just quickly tell you about what we do, since you do not know. We work in the area of computational biology, bioinformatics, biomedical data analysis, complex systems modeling, and specifically food data analysis. This is the lab webpage. You might want to look at it for getting more information. One PhD position definitely is going to be aligned with a recent discovery that we have done about a special pattern found in the case of Indian cuisine, how recipes are combined together, and these are the various domains associated with it. Algorithm development for creating a novel recipe recommender system for novel recipe generation. That's one of the projects. And there are hypothesis-driven projects around food data. and that's one of the PhD problem aligned with. Although the other projects, summer internship that I'm talking about, they will be, some of them, 2 of them will be in these topics. Okay. Broadly speaking, we work on modeling biological complex systems. The idea is to model, control, and hopefully learn design principles. In the area of protein structure, basically structural bioinformatics is one topic of interest. Disease modeling using interactome data is another topic on which we work on. Target identification and in silico drug discovery. And partly even the in silico drug discovery goes in the direction of bridging modern and traditional medicine. That's a broad topic in which we're working on. Not all of these areas we have PhD positions open, but internships, definitely there are threads which are open in this area. And finally, data analytics, one of them specifically on which one of my PhD students is already working, Rakesh, is about side effect predictions using drug target and side effects information. So that's broadly the overview of kind of work we are doing around the theme of asking questions, hypothesis-driven questions, designing tools, and databases. So that's a broad theme. Any of you who are still interested, you can talk to me today or you can write an email as said. Alright, so thank you. Thanks for listening. We are done with the theoreticals and the lecture sessions now. We are moving on quickly for a photograph session followed by the lunch. I would expect you to finish the lunch as soon as possible because you would take time to settle down in the laboratory. So I suggest a time of around 2:10, half an hour. Okay, so 2 o'clock, ideally speaking, you should be moving on soon after finishing the lunch to the lab sessions. Thank you.